Point mutations represent one of the most fundamental types of genetic changes. These are alterations that affect just a single nucleotide in the DNA sequence. Despite their small scale, point mutations can have significant effects on the organism, ranging from no impact at all to causing serious genetic disorders. A point mutation is defined as a change in a single nucleotide within a DNA sequence. Remember that DNA is made up of 4 nucleotides: adenine, thymine, guanine, and cytosine. In a point mutation, one of these nucleotides is replaced by a different one. This is the smallest possible change that can occur in DNA, affecting just one building block out of millions. Point mutations are classified into 2 main categories based on the chemical structure of the nucleotides involved. A transition occurs when one purine is replaced by another purine, such as adenine to guanine, or when one pyrimidine is replaced by another pyrimidine, such as cytosine to thymine. A transversion occurs when a purine is replaced by a pyrimidine, or vice versa. Transitions are more common than transversions because they involve nucleotides with similar chemical structures. Let's look at a specific example of a transition mutation. In the normal DNA sequence, we have adenine, thymine, guanine, cytosine, adenine, thymine. In the mutated version, the adenine at position 5 has been replaced by guanine. Both adenine and guanine are purines, so this is a transition mutation. Notice how just one nucleotide change can alter the genetic code. Now let's examine a transversion mutation. Starting with the same normal sequence of adenine, thymine, guanine, cytosine, adenine, thymine, the mutated version shows the adenine at position 5 replaced by cytosine. Since adenine is a purine and cytosine is a pyrimidine, this represents a transversion. This type of change is less common but can have different effects on the resulting protein. Point mutations can have 3 different effects on the proteins they encode. A silent mutation results in no change to the amino acid sequence due to the redundancy of the genetic code. Multiple codons can code for the same amino acid, so the mutation may not matter. A missense mutation causes a different amino acid to be incorporated into the protein, which may alter its function. A nonsense mutation creates a premature stop codon, leading to a shortened, usually non-functional protein. This is often the most severe type of point mutation. Point mutations arise from several sources. DNA replication errors occur when DNA polymerase occasionally incorporates the wrong nucleotide during DNA copying. Environmental mutagens, such as ultraviolet radiation, chemicals, and certain viruses, can damage DNA and cause mutations. Spontaneous chemical changes happen naturally, such as the deamination of cytosine to uracil, which can lead to mutations if not repaired. These processes constantly introduce small changes into our genetic material. To summarize, point mutations are single nucleotide changes that can have varying effects on organisms. They can be classified as transitions, involving similar nucleotides, or transversions, involving different types of nucleotides. Depending on their location and the genetic code, these mutations may be silent with no effect, missense causing an amino acid change, or nonsense creating a stop codon. Understanding point mutations is essential for comprehending genetic variation, evolution, and inherited diseases.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.